In this objective, we're going to be looking at the disaccharides sucrose, lactose, and maltose. We're going to need to know what each of those disaccharides is made of in terms of the monosaccharide monomers that are put together in order to create that. So this objective number 10, is on the disaccharides, sucrose, lactose, and maltose. So these disaccharides, sucrose is glucose plus fructose, lactose is glucose plus galactose, and maltose is glucose plus glucose. In each case, we're going to need to know the two monosaccharides that make that disaccharide. That's just a pure memorization problem. We're also going to need to know where the dietary sources are, and the names tell us what the dietary sources are. So if we know that sucrose is table sugar, we know what the dietary source of that is, for example. Disaccharides are formed by something we're going to look at in detail later on, which is what's called a dehydration synthesis. A dehydration synthesis is one of the only chemical reactions that we're going to need to learn in this course. There's only two of them. One is the formation of the disaccharide bond in a dehydration synthesis. The only other reaction that we're going to need to worry about is the hydrolysis reaction, which consists of breaking these things apart. If we put them together, we expel a water molecule as a result of putting together glucose and fructose. That's shown in the green oval here. When we expel the water molecule, then we form an oxygen link between these two monosaccharides, and now we have the disaccharide sucrose. We've expelled water, so we're going to call that a dehydration synthesis. If going the other way, from right to left, we use water to break something. Water is hydro. Lysis is the breaking of something, the breaking apart of something. We're using water to break chemical bonds. And in this case, the water is going into the structure to help take it apart. And when we add water back to this sucrose in a hydrolysis reaction, then we get back out glucose and fructose. So the dehydration synthesis is where we put these two monomers together into a disaccharide. When we do the hydrolysis, we're taking apart this disaccharide and making two monomers, or two monosaccharides. So far, we've looked at the monosaccharides, the pentoses and the hexoses. So we had ribose and deoxyribose as pentoses, and we had glucose and fructose and galactose as hexoses. We put those together in different combinations and made disaccharides. So we had the disaccharide sucrose, we had the disaccharide lactose, we had the disaccharide maltose. Now we're going to string them together into even bigger combinations, and those bigger combinations we're going to call polysaccharides. So the next objective, objective 11, is to look at polysaccharides. So remember that the monosaccharides are the structural subunit, that when we string them together into a polymer, we get a polysaccharide, polymer, polysaccharide. We're joining these sugars together, recall from the last objective, by dehydration synthesis. In plants, we form long strings, either branched or unbranched, of these sugar monomers, of these monosaccharides. So in plants, we call these cellulose or starches. Cellulose is what makes up the walls of cells. That's where it gets its name. Remember that anything which is a carbohydrate is going to end in OSE. So one thing we need to recognize throughout this course is that if a chemical name ends in OSE, then it's a carbohydrate. So notice that the monomers, the monosaccharides, the disaccharides, and the polysaccharides all end in OSE because we're describing carbohydrates. Cellulose is a carbohydrate, ose, which makes up cell walls. It's non-digestible, so on the nutrition label we're going to call this fiber. The only way that human beings can digest cellulose is with the help of microorganisms living in the gut. But human beings are very good at digesting starches. Starches, for example, in potatoes and other starchy foods, are a storage form of glucose and a major source of carbohydrates in the diet. The plants are storing starches for their own energy needs, but humans can take those starches and break them down for the human's energy needs. When a human breaks down glucose into its monomers, absorbs them through the digestive tract, dumps them into the bloodstream, and then they're eventually carried to the liver and to the muscles, where they're turned into a polysaccharide that's called glycogen. Glycogen in humans and other animals is the storage form polysaccharides. So we put together these monosaccharides into a storage form that's a polysaccharide. This gives us a ready source of glucose if we need glucose suddenly. For example, if I run a marathon, then I need to carbo load before the marathon. The process of carbo loading is the process of putting as much glycogen as my physiology can possibly hold in my liver and muscles, and then using that glycogen, breaking it down during the race to run as far as I can before I hit the wall, using the stored glycogen that I've put in my liver and muscles. When I run out of glycogen, that's, as I described earlier, that's what runners call hitting the wall. Bicyclists do the same thing at roughly 75 to 80 miles into a century on a bicycle. In runners like me, we hit the wall at mile 18, mile 20. That's the point at which our glucose storage form as glycogen has completely run out, and we have to rely on other carbon sources for our energy. Unfortunately, that process is not very efficient, and so it's not a very pleasant sensation when you hit the wall. Glycogen is the major storage form of glucose in humans. As I said, mostly in liver and muscle. The liver is able to store 70 to 100 grams of glycogen. That 70 to 100 grams can be converted to about 280 to 400 food calories or kilocalories of food energy. It takes about 100 calories to either walk or run a mile. For that same 100 calories, you can bicycle about 4 miles. So bicycling is about 4 times more efficient in terms of moving you around as walking or running. In muscle, we can store 200 to 400 grams of glycogen depending on our physiology and the makeup of our muscles. That's 800 to 1600 food calories or 1800 to 1600 kilocalories in energetic terms. If we add that together, then the most amount of glycogen that we can store is typically around 2,000 calories. And remember, I said it's about 100 calories, food calories, per mile of running or walking. So you can go about 20 miles before you hit the wall. And those of you who are runners will recognize that as being the point at which most people have used up all their storage of glycogen and have hit the wall. So glycogen is used because we can easily break it down to glucose, which, as I said earlier, is the preferred energy source for cells. So glucose is the main kind of energy that's found in blood. It's the main carbohydrate in blood. So just like plants had starches and cellulose as polysaccharides, putting those monosaccharides together into big, large, branched or unbranched chain structures. In humans and other animals, then we use the form glycogen as a source of glucose. The last application of the carbohydrates that we're going to see is in the process of forming polysaccharides that are used as cell surface markers. We think of these as cell surface flags showing what the identity, for example, of a cell is. For example, we may know that on red blood cells there are different red blood cell groupings. And these different red blood cell groupings exist because of the existence of different polysaccharides on the surface of the cell. So shown at the top of this slide is blood type A, and it's not important for us to know the sugars that go into this. In fact, the sugars, some of them are going to have names that are unfamiliar to us that we haven't studied yet in this course, and we won't study unless we get to a biochemistry course. But blood type A, blood type B, and blood type O, as we can see here, have different combinations of sugars in different arrangements that the other cells of the body, particularly the immune cells of the body, recognize as either belonging to us or belonging to someone else. This is the basis for blood grouping and why it is that certain blood groups can only be transfused into other blood groups. We'll talk about that in the Health Sciences 1111 course when we study blood. As I said, some of these names are unfamiliar, but you'll notice that these are all sugars, some of which we've seen before. We certainly know what galactose is, and we've studied that before, and that's at several places here. We haven't studied the modified sugars in acetylglucosamine. We haven't studied the sugar fucose. And so those are also in this. We don't need to know this for this course, but this is just showing you that by putting these sugars together, these same three sugars together into different arrangements, that we end up with different blood groups. All the red blood cells of our body have whatever blood type is characteristic of us. So if we have blood type O, as I do, then all of the red blood cells contain this bottom structure on their cell surface, identifying them as red blood cells of blood type O. In addition, if we have blood type A, we not only have the blood type O structure on the surface of our red blood cells, but we have the additional structure of blood type A shown at the top here. If we have blood type B, then we have both the type O structure and the type B structure on the surface of our cells, of our red blood cells. And if we have blood type AB, then we have all three of these structures on the surface of all of our red blood cells. That's what makes us blood type AB. So all red blood cells, regardless of what blood type you are, have blood type O marker on their cell surface. People with blood type A have both the A and the O polymers on the surface of their red blood cells. People with blood type B have both the B and the O polymers on the surface of their red blood cells. And people with blood type AB have the A, the B, and the O polysaccharide chains on the surface of all of their red blood cells. So just to sum up what we know already about carbohydrates, what we've learned throughout life, and also what we've added onto that knowledge in this section of the course, carbohydrates are important for a lot of different processes. We've talked about some of those here. Why are carbohydrates so important? Well, they give us energy-yielding nutrients, for example, starch from plants. They give us building materials. So cellulose, because it's not broken down, is used in wood construction, paper, cotton-based clothing, and cell walls in higher plants. And then water-soluble molecules are carbohydrates oftentimes. And so the mono and disaccharides that are used as sweeteners in some of the foods that we eat. Well, carbohydrates are typically an important part of the human diet. So most humans with a balanced diet will eat about 2/3 of the calories that they take in through carbohydrates. They can be used for energy, as we'll see later on in Module 4, under aerobic and anaerobic conditions. So whether we have lots of oxygen available or we don't have oxygen available, we use carbohydrates for energy. And so about 1/6 of the calories in our diet will come from proteins, about 1/6 of the calories in our diet will come from fats, but about 2/3 of the calories in our diet will come from carbohydrates. So as we can see, we really need to know a lot about carbohydrates because they're an important part of the diet. They're also an important part of the constituents of our body. So this concludes our study of the carbohydrates, the first of the 4 classes of carbon-containing or organic molecules that we're going to study in this course.